The purpose of this presentation is to answer the following question: does stress impact the GI system? So this presentation will go over an overview of the GI system in whole, 5 summarized articles that we have read about different stress-induced GI issues, and then we'll circle back to this question and answer if it truly does impact the system. Process of the GI system. There are 6 major processes, and it starts with ingestion, which is eating. Then we have secretion, and secretion in the digestive system refers to the release of enzymes, hormones, and other substances that help the body digest food. Now we have propulsion, which is moving food throughout the body. We can do it by swallowing, which is done in our oropharynx. And it can also be done by peristalsis, which moves food along the tract by relaxing and or contracting muscles in our organ walls of the esophagus, stomach, small intestine, or large intestine, depending on where it is in that moment. Then we have digestion. There's 2 types. We have mechanical and chemical. So in mechanical, we have it prepares for digestion by enzymes by either chewing, mixing our food with saliva, churning food in the stomach, and segmentation. And as for chemical, enzymes secreted into the lumen break down complex food molecules into their chemical building blocks so it can digest. Then we have absorption, which is when the passage of digestive products go through the mucosal cilia by active or passive transport into the blood or lymph. And finally, we have defecation, which eliminates indigestible substances from the body via the anus in the form of feces. The enteric nervous system will be helpful in understanding the brain-gut axis and how stress relates to the GI system. So this is like the CNS of the brain, but for the GI tract, and it has over 100 million neurons making connections. Enteric neurons involved in the submucosal and myenteric nerve plexus, which regulate digestive activity and provide major nerve supply. The submucosal plexus innervate secretory cells of the mucosa and submucosa, which are important for hormonal controls of the GI system. Then our myenteric plexus innervates smooth muscle of the mucoclerus externa, which is important for GI motion. So these 2 things are very ideal in our body's hormonal control and motion for the GI system. Because of the ENS, it allows the GI system to have reflexes. They're short reflexes which control the pattern of segmentation and peristalsis, which is largely automatic, involving pacemaker cells and reflex arcs. In these reflex arcs, we have sensory or motor messages. So first we'll start with how sensories work. They have mechanoreceptors, which detect distinction due to the entry of food. And then we have chemoreceptors, which involve osmolarity, changes in pH, and products of digestion. Then we have the motor messages, which are short reflexes that are independent of the CNS and regulate localized activities affecting small segments of our GI tract. Our long reflexes are where the ENS sends information to the CNS via visceral sensory fibers. These long reflexes can be initiated by stimuli arising inside or outside of the GI tract. In these reflexes, the ENS acts as a way station for the ANS, allowing intrinsic controls to influence digestive activity. So we can have our parasympathetic, which is the rest and digest, which will promote digestive and absorptive behaviors in our GI system. Or we can have sympathetic, which is our fight or flight. So that will shut down these activities that the digestive system provides for our body. This first article addresses stress in the brain gut axis and functional and chronic inflammatory gastrointestinal diseases. The paper looked at the tri-social stress test, which is a standardized psychosocial trigger of acute stress responses. It induces transient increases in psychological and biological stress markers, including heightened arousal and state anxiety, as well as activation of the HPA axis and the sympathetic nervous system. There's other experimental protocols such as auditory stress, sad music, watching fear-listing movies, and they've been applied to visceral sensory-motor functions in healthy individuals and patients with FGIDS and IBD. So, found acute stress impacts perceptual mechanisms, GI motor functions, local inflammatory processes, and gut permeability, which are all important in understanding the inflammation in IBD. In healthy volunteers, acute pain stress induced musical mast cell activation and epithelial damage. Stress-induced musical mast cell activation and epithelial damage are more pronounced in IBD patients than in healthy controls. Acute stress effects vary depending on the gastrointestinal segment and stress type, intensity, and duration. Psychopharmacological approaches such as CRH have been emerged in experimental GI research to target stress-related neuroendocrine mediators. The study found that elevated cortisol levels increase visceral pain sensitivity in healthy individuals, especially in women. Public speaking stress leads to an increased intestinal permeability, suggesting cortisol may be crucial to stress-induced changes in peripheral mechanisms regulating visceral afferent signaling and pain processing. Chronic stress and high comorbidity with affective disorders, particularly anxiety and depression, are considered risk factors for the development and maintenance of GI inflammatory disease. The symptoms of stress correlate with the severity of GI symptoms, leading to reduced quality of life and altered illness experience and behavior. Psychological factors contribute to or explain alterations in the brain function or structure observed in patients with IBS and IBD. Prospective cohort studies have shown that impaired quality of life and increased anxiety and depressive symptoms are independent predictors for the first manifestations of IBS. High levels of these perceived stress are linked to rectal mucosal abnormalities in patients with IBD. It enhances gut inflammation and triggers more intense symptoms. A possible limitation could be the transdisciplinary approach activated by the authors is valuable for fostering collaboration and integration across different fields of study, but it may also present challenges in terms of merging conflicting findings or interpretations from different disciplines. This article showed through the psychological stress in rats that it can lead to GI barrier dysfunction, even though stress of rats during shipping and handling increases GI permeability. The main mechanism is the release of glucocorticoids, acetylcholine, corticotrophin releasing hormone, and muscarinic receptor activation, intestinal mast cell activation, and reduced blood flow due to sympathetically driven splanchnin vasoconstriction may also contribute to this dysfunction. But what does that mean? So the GI barrier dysfunction is a breakdown or impairment in the integrity of the lining of the organs and the intestines. And when this barrier becomes compromised, it can lead to increased permeability, as well as we can see that the stress increase the permeability already, allowing harmful substances like bacteria, toxins, and undigested food particles to leak through the intestinal wall and enter the bloodstream, which will lead to all sort of compromised immune system, diseases, and things. An intervention, the article does say, is glucagon like peptide 2 is an intestinal trophic growth hormone, and it has been shown to reduce chronic psychological stress-induced intestinal barrier dysfunction. However, a limitation to this paper can be attributed to the usage of rats instead of humans to analyze this issue. Next, we have the psychological impact that stress has on people with IBS. So, symptoms of IBS are lower abdominal pain or discomfort associated with altered bowel habits, especially constipation or diarrhea, can be seen. This paper explored the impacts of stress on inducing IBS and found musical mast cells are activated after acute stress and release neuropeptides, which are responsible for the altered intestinal sensation, motility, secretion, and permeability characteristics of IBS. Early life stress also impacts the central nervous system and peripheral nervous system, leading to altered visceral hypersensitivity and IBS. The strong linkage between psychological stress and IBS originates from the brain-gut axis, with the ENS playing an essential role in regulating gut physiology. Stress can be the etiology for the development of IBS or aggravation of symptoms, and can be exacerbated psychological disorders and lower health-related quality of life. So some reasons why this might affect it is the role of CRF signaling system. So stress affects the composition and growth of microbiota, maintaining bidirectional communication between the brain and gut axis. And microbiota is important for when bile is transported by the liver to the small intestine for the purpose of breaking down fat and gut flora to help metabolize. Bile acids are broken down by bacteria, which enables the liver to recycle them and keep the blood cholesterol levels from rising. Microbiota also plays a part in communicating with the CNS through the brain-gut axis. Another reason is the stress on the brain-gut axis. It stimulates HPA axis and triggers the release of CRF, ACTH, and cortisol, which directly or indirectly affects gut function. And stress alters the quality of mast cells, EC cells, lymphocytes, and their produced neurotransmitters, which are involved in musical and immune activation and further interact with gut microbiota and gut function. So the impacts of the gut-brain axa and the gut microbiota contribute to IBS symptoms. A limit to this article is also a heavy reliance on animal studies to support the conclusions about the relationship.
This article talks about the effects of occupational stress on the GI tract. And the goal of this article is to review and to provide an overview of stress and its negative relationship with the GI tract. So occupational stress can affect the stomach and intestine, leading to various digestive issues. The Internal Labor Organization recognizes the importance of mental well-being in the workplace. Current definitions of job-related health risks often overlook psychological stress. Despite this, many studies have explored how the stress impacts the body, with a focus on jobs deemed as stressful. Among the physical efforts of stress, digestive problems are common. In everyday medical practice, doctors often seek a link between job stress and digestive diseases. However, the usual approach is to treat the symptoms rather than the root cause. Employers also tend to underestimate the role of stress causing these problems, and it's vital to identify, measure, and manage the risk in the workplace, and to recognize it as a significant health risk. Personally, for me, what comes to mind as someone in nursing school is just thinking about the stress of being a nurse and everyday challenges and just the amount of time spent at the hospital and the long shifts and the stress that can come with that. But workers who experience a mix of anxiety, depression, and stress are more likely to have digestive issues. Certain types of jobs, like those involving deprivation, fatigue, and chronic anxiety, are particularly associated with conditions like ulcers, IBS, and indigestion. The body's autonomic nervous system, hormonal system, and genetics all play a role in how stress affects the digestion. Understanding and addressing these factors are essential for promoting better workplace health and better digestive health.
A limitation to this article can be how it focuses on a specific population, individuals experiencing occupational stress. The findings might not be directly applicable to other populations or contexts.
Next, I'm going to talk about the role of stress in functional GI disorders. So there's evidence for stress-induced alteration in GI motility and sensitivity. Psychological stress is believed to significantly impact functional GI disorders. IBS causes worsening symptoms with stress. So studies consistently demonstrate that acute or short-term stress leads to slower gastric emptying and the colon moving faster. The alterations in GI motility are thought to contribute to symptoms such as changes in stool, frequency, and consistency. People with IBS tend to have an even stronger reaction to stress, especially in their colon, possibly due to being more emotionally reactive. Serotonin is released in response to stress and makes the colon move faster by activating certain receptors. A substance in the brain called CRF plays a role in how stress affects our gut. It slows down stomach emptying and speeds up colon movement through specific receptors in our body. Stress can also make the digestive tract more sensitive, especially if it causes big emotional changes. This sensitivity is controlled by CRF in the brain and involves areas that are linked to emotion. As well, mast cells become more active when we're stressed, which can also make our gut more sensitive. Stress affects how our GI tract works and how sensitive it is through complex interactions involving CRF, serotonin, and emotional factors. More research is needed to fully understand these processes and improve treatment for GI disorders.
And a limitation to this article is how there's evidence from experimental animal studies and some limited data from investigations in healthy subjects in patients with functional GI disorders. There is still a lack of comprehensive understanding regarding the exact mechanisms. So, finally, looking back on our original question that we wanted to figure out, does stress impact the GI system? And from the research articles and from our knowledge of the GI system, yes, it does impact our GI system function. As mentioned earlier, human stress affects our intestinal permeability, which allows toxins, bacteria, and other harmful substances to leak into the bloodstream, triggering immune responses and inflammation in the GI tract. Composition and growth of microbia is impacted by stress. Microbia assists in the digestion of complex carbs, fiber, and other dietary components that the human digestive enzymes cannot break down. Sternobacteria produces enzymes that can break down otherwise indigestible substances, releasing nutrients that can be absorbed by the body. The gut microbiota helps maintain the integrity of the intestinal barrier, which serves as a protective barrier against harmful substances and pathogens by promoting the production of mucin and enhancing the tight junctions between intestinal cells. The microbiota helps prevent the leakage of toxins and pathogens into the bloodstream and many more. And as we talked about earlier, the microbiota gets impacted when stress is in the picture. So all of those functions will be impaired, which is not great for the function of our GI system. And looking back at our ENS, our brain gut axis, if it's impaired, we know that the parasympathetic system is responsible for rest and digest, and it promotes digestive and absorptive activities. And we know the sympathetic is responsible for our fight or flight. So, if we are experiencing stress, aka our fight or flight, our digestive system is gonna be shut down and impacted, and things aren't going to be running properly. As well as if our parasympathetic system gets impaired by our stress, our rest and digest is not going to be happening. There will be no promotion of that happening. And so it's not going to function well. And on top of that, the stress can also lead to diseases or additional symptoms of diseases humans already have, such as the IBS and the IBD. So it truly does impact our GI system.